What is a cell? Basic, living, structural, and functional units of all organisms. Life is composed of cells. Now, you can be unicellular, or you can be a multicellular organism. Humans are multicellular organisms. We have different cells doing different tasks, but we're one organism. This comes from the Latin cella, meaning chamber or storeroom. Whenever they were looking at the first cells under microscopes, they were looking at cork or plant cells, and these looked like little chambers that monks stayed in the monasteries. So when we look at a cell and we want to say, okay, well, what makes up a cell? Well, what separates a cell from anything else in the world? That limiting boundary that we talked about as a life processy earlier in the unit. So the limiting boundary of a cell is a plasma membrane. So we know cells have plasma membranes. Remember, this is this limiting boundary of the cell is the plasma membrane, and that's one of the life processes. So anything that's alive has an outer boundary. Anything that's alive separates itself from its external environment. A cell does that through the plasma membrane. So we're separating inside, outside. If you want to go into a cell, you got to go through a plasma membrane. If you want to leave a cell, you got to go through a plasma membrane. Okay? So the other thing that we have are organelles. These are things like smooth ER, rough ER, Golgi apparatus, lysosomes, mitochondria, and we'll break down each one of those. So an organelle is a specialized structure that has specific functions. So if I do a different color, we're going to say specific functions. Each organelle has a different function. This is going to allow for a division of labor within the cell, which means different parts can be doing different things at different times. So you don't have to switch all the cell on to start making ATP. We just let the mitochondria take care of that. So cells can do multiple things at one time because different organelles do different things. The last thing that we have is the cytoplasm. And the cytoplasm is that intracellular fluid of the cell, but it's not just fluid, it's like a gel-like substance, right? Because it's not just the cytosol, which is the fluid component of the cytoplasm. We'll look into that more. So anyways, cytoplasm, intracellular fluid. That's what we'll say for now. So this guy, the plasma membrane, is separating the inside and the outside of the cell. That's a life process called a limiting boundary. Second thing, these organelles within the cell are doing specific functions which allow for a division of labor. A division of labor within a cell. So you can have different tasks going on at the same time. And then we have the cytoplasm, which is going to have all our organelles and some other stuff. All right. So the fluid mosaic model. A mosaic is, this is a definition from Wikipedia, a picture or pattern produced by arranging together small colored pieces of hard material such as stone, tile, or glass. So let's put that into some science terms. Let me read it again. A picture or pattern produced by arranging together small colored pieces of hard material such as stone, tile, or glass. So instead of using an art concept, we want a biological definition. So we'll say it's a mosaic. The plasma membrane is a fluid mosaic because it's made up of a bunch of different parts. So it's a mosaic because it has all these tiny little different parts in one thing, making up this plasma membrane. It is fluid because you don't want to have this idea that things are staying stagnant in a plasma membrane. So if I have a plasma membrane here, and within this plasma membrane, I have an integral protein. This integral protein goes through the entire plasma membrane, access to both the intracellular and extracellular fluid, and things are passing in and out of this guy. We can't think that this is going to stay there forever. In a second, it might be way over here, and then in another second, it might be way over here. So if I drew a cell, and this is its plasma membrane, and we say that we have an integral protein here, well, next time we looked at the integral protein, maybe it's over here now. So it's fluid because this moved from here to over here. All right. So these different components within the plasma membrane, they're not stuck, they're not static, they're not rigid. They're constantly moving around, and that's what gives us this fluidity and this mosaic made of a bunch of different stuff. Okay. So for that example, just because a protein's integrated in the membrane, it doesn't mean it's stationary. It can be in one place and then fluidly move around. Okay. Let's look at the composition of the plasma membrane. Let's look what it's made up of. So I'm going to write what it's made up of first. So these are components of plasma membrane. I'll write them all up, we'll talk about them, then we'll draw a picture to put it all together. Okay, let's think about this. We got our plasma membrane. We should know by this point the plasma membrane is a double bilayer of phospholipids. So if you look back through our other video, we talked about how a phospholipid is an altered triglyceride that's removed a fatty acid tail, added this phosphate head, which gives it hydrophilicity, meaning that it likes water. That's the polar head. And then this nonpolar hydrophobic tail, this fatty acid tail, that's going to be pointing inward. So this bilayer is formed like this. We have these different heads, and then we have our tails oriented towards each other. So go back and watch that video on polarity and all that if you need a recap on the polar heads versus the non-polar tails. But we know that water is polar, and these heads are polar, so the heads are oriented towards the extracellular fluid and the intracellular fluid. All right, within this guy, we also have these integral proteins. So integral proteins pass through both sides of the plasma membrane. Okay, that means that things going or interacting on one side of the integral protein can probably go through this integral protein and interact with the other side of the plasma membrane. We'll just draw them. We'll draw them in a few minutes. Peripheral proteins. These are associated with one side of the plasma membrane, so maybe they're attached to an integral protein or maybe they're embedded into the plasma membrane, but they give structure to the plasma membrane, and we'll see how else they're used later on. Both integral and peripheral proteins can be activated to change the activity within the cell. We'll show some channel proteins in a minute. So cholesterol, this acts like glue, so we'll just call this the glue of the plasma membrane. It's going to hold things in place. Okay. Next is carbohydrates, and I'm actually going to, we'll just put for now we'll just put, these are linked monosaccharides. So our monosaccharides were glucose, fructose, and galactose. And carbohydrates can play along with these glycoproteins. And remember, glyco looks like glucose, right? Looks pretty similar. So this has to do with sugar and protein. So this is a carbohydrate attached to a protein. And this is involved in cellular reception. Glycoproteins. So we're on these plasma membrane functions. I have them all written up. So we're going to talk through them and try to identify which components that we just talked about of the plasma membrane are doing each of these functions that he discussed in class. So the first one. Separates internal, external environment. Man, that sounds a lot like what we thought of with this limiting boundary of the plasma membrane. So really, I mean, everything is acting as the limiting boundary because the phospholipid isn't the only thing that's making up that plasma membrane. But I really like to think of phospholipids because they're the main component of this plasma membrane. Facilitates contact with other cells. That's a glycolipid. We already said that. Cell-to-cell communication, important in immunity. And then foreign substances. Well, we can say glycoprotein right there. And maybe glycolipid for that one too. Provide receptor sites. We already said glycoproteins and carbs can play that role. And then control flow in and out of cell. We'll just we'll leave that blanketed because there's a few things that affect what's coming in versus out, and we'll talk about that as well. So these are the four functions. Separates internal, external environment. If it's the internal and external environment, then it's probably the thing that interacts with that external environment, right? That's the second bullet point. Third, if it's going to interact with this environment, we have to have a structural component that allows us to do that. That's a receptor site. And then things go in and out of the cell. So you're not leaving a cell unless you go through the plasma membrane. You're not coming into the cell unless you have a plasma membrane. Some things can pass through freely. Some things need help. We've talked about this idea of polar and non-polar, or you can use the words water-soluble and lipid-soluble. So if we're looking at the plasma membrane, non-polar, which is also named for lipid-soluble, pass freely through the plasma membrane. Water-soluble molecules, we either need a receptor, a channel, or some other mechanism to get us through. Okay, we'll talk about those. Now, I said we're basically going to expand upon this controls the flow in and out of a cell. So what are some things that are going to determine the function of the plasma membrane? What's going to determine whether things can go in and out of the cells?